Alors, je présente ce événement de RiSBOd. RiSBOd est une organisation pour unifier tous les étudiantes, mais aussi les autres partenaires de RiSBOd, les autres étudiantes des autres universités de McGill, Université Laval et de UDM. Et un de les rôles, c'est de partager toutes les connaissances scientifiques et les autres choses, et de unifier tous les membres, les autres personnages de la monde. So RiSBOd, one of the goals is to unify students and share knowledge. And one of the very interesting topics that we have today is something that touches me in my life. It's cancer. Un des topics qui est très proche à mon cœur, c'est le cancer parce que euh, ça a vraiment affecté ma famille beaucoup de fois. It's something that is really dear to me. And if there are ways to control cancer, perhaps through epigenetics, it's important to realize or think and ponder about these things. S'il y a une façon pour contrôler le cancer, c'est vraiment important. Euh, et maintenant, on a un conférencier invité très spécial, Federico Granados Unger, qui a de recherches sur ça, sur le cancer et les épigénétiques. We have a guest here today who has focused on research in epigenetics and cancer. Et avant de ça, il va partager quelques informations de sa vie avant de faire la recherche. He'll also share some information for which he's done before starting research. And I thank you all for being here. Et je remercie tout maintenant. If you are ready, Federico, you may begin. And thank you so much for being here.
Sure, thank you. Can you allow me to share screen, please?
Yes, absolutely.
Permission. Thank you very much. And yeah, first of all, yeah, thank you for the invitation, Michael. I'm very honored to be here talking about a very fascinating subject and with great possibilities for the future for cancer researchers in Canada and abroad, all over the world. And yeah, it's related to my research during my master's degree right now. So I'm very happy and to be able to share a little bit about that and hopefully inspire some of you to pursue this line of research if you're in graduate studies or looking forward to. So yeah, so first, yeah, thank you, Michael, as I mentioned, also RiSBOd and the McGill Faculty of Dental Medicine and Oral Sciences for inviting me to share a little bit of my lab's research focus, and also a little bit of my background, which might be well inspiring and interesting for the audience. I mean, before research. So yeah, so just a couple of minutes to give a little bit of insight to my background. So before coming here to Canada, to the University of Montreal, where I'm currently pursuing my master's degree in pharmacology at Dr. Reynald's lab. So I was always I have always been very passionate about space and space research. In fact, well, during my bachelor's in biotechnology engineering, which I studied back in Mexico, and also during the pandemic, I started participating in something that's called space analog missions, which are like simulated space missions. Like, for example, if somebody here on Earth wants to, in the future, go to the moon or Mars, right, and perform some scientific experiments there or build a colony there. First, we simulate that on Earth to see what social factors or what advancements to our current technology we need to do before we go there, right? And so I participated in a virtual, obviously because of the pandemic, mission at here one analog space station called Habitat Marte, which started my career in space, which goes hand in hand with my experience in cancer research, which I also pursued just before the pandemic at a research internship at the McGill Hospital, McGill University Hospital Center, in 2019. But so yes, I've been like jumping through in like doing skydiving, doing some hiking, rock climbing in Mexico in preparation for something that's called career astronaut competition, which is organized by an organism called Advancing X, located in the United States, which gathers people from all over the world, and you enter a competition, 5-year competition, where they present different challenges to the selected candidates, and the final prize is a suborbital flight, right, to like those carried out by Virgin Galactic. So in here, in the last two images here in the center, like bottom and bottom right, it's me like doing some rock gaining and scuba diving challenge. Well, that was actually after the challenge as part of this competition. So yeah, I've been very involved in the space sector so far, but at the same time, or well, afterwards, continuing that initial research experience at McGill University, I decided to just pursue full-time this parallel passion, which is related to cancer research. So I started my studies in 2023 at the center San Justin, the hospital, the research center of that hospital here in Montreal. And so there I've been very busy in, also research conferences, looking at creating a new three-dimensional model of one type of lung cancer, which is called non-small cell lung cancer, which is actually one of the most lethal with respect to like caused by cancer, like the main reason of death here in Canada and in the world, right? With millions of people who lose their lives due to this disease. So what we're trying to do, what I'm trying to do in my research, which I'm finishing right now, I'm wrapping up with my thesis, is trying to develop this three-dimensional model, which we call spheroids, which you can see here at the bottom right. This is a cross-section of a lung cancer spheroid stained with some specific markers that were of interest. So that's what you see on red. The blue dots are like the cell nuclei. And so we're trying to make this model because we have seen in our lab that the current models that are used, that is in 2 dimensions, where they only use cancer cells to test new drugs, doesn't predict whether very precisely how which drugs will work in patients. We think that the reason of that is that we are lacking a three-dimensional model, like the tumors. Like tumors in people are three-dimensional, and so we're trying to do this model so that and analyzing what like pathways, signaling pathways, or what is the response to certain chemotherapy drugs, and compare that response at that signaling specific to our models to real patient tumors. And once we validate that our model is very similar to patient models, we want to push for labs all over the world to use three-dimensional models for everything, basically in the research, because I think this is the way to go, and this will save a lot of lives and money in the long term. So yeah, that's my background so far. And my lab, we focus a lot on epigenetics. And well, if you came into the stock, maybe maybe you're not very familiar with this term. So I'm gonna give a brief introduction about epigenetics and epigenetic mechanisms to those in the audience that are not very familiar with it. She's fascinating. So what are epigenetic mechanisms? So these are changes that are heritable. This is very important. We'll come back to that later. Changes in the expression of genes that are independent of the DNA sequence. So this means that these are not somatic mutations like the point mutations, for example, or translocations that you might see talked a lot about when researchers say, oh, there were some mutations in the cells of this patient which caused cancer. So those are more related to the somatic mutations. But this talks about something that is outside of that, right? It's above that. EPI means above, so above genetics, right? It's more like the switches which control which genes in your body are turned on, are turned off, right? And as you may start to think, well, if you can turn on and off these mechanisms, then maybe you can control a lot of things in the human body and at will, right? So that is the fascinating thing about epigenetics. The one important thing that is very striking for me when I first thought about epigenetics is that, well, all of our cells in the whole body have the same DNA, exactly the same DNA. But what determines whether one cell is a brain cell, a lung cell, muscle cell, bone cell, um, it's not the genetics, but they're like the epigenetics, those mechanisms that are above the DNA that are able to tell each cell what is that cell gonna be, right? So just these changes in the compaction of the DNA determine the cell fate. So as you may start to see what is the importance of epigenetics and how wide its effects must be, right? Which is really cool. So what are these epigenetic mechanisms? So there are four main epigenetic mechanisms to alter the expression of certain genes in the body. And so the ones that we're going to focus more today are the first two. Well, related to the third one also. So the first one is the methylation. So the addition of methyl groups on the cytosine nucleotides of the DNA that precede a guanine, right? So we call these CPG islands, which are regions in the genome that are full of cytosine and guanine, like in repeats or like with a high concentration of these two bases. And the second one is post-translational modifications of histones on certain amino acids. So, well, we're gonna see what histones are in more detail afterwards. But just know that these are a little chains of amino acids that come from something called the nucleosomes, which is like the little bits that conform something called chromatin. Chromatin is here, as you can see in the image in the left, like DNA plus these proteins. So these are used like to really compact the DNA so that it can fit in each cell nuclei. But we will move to that in more detail later. And so these modifications, we call it histone acetylation. So the addition of acetyl groups to certain amino acids of these little protein tails that come out of these nucleosomes. Histone methylation, the addition of a methyl group, phosphorylation, the addition of a phosphate group, or ubiquitination and sumoylation, which are other types of modifications you can have here. Then the third one is the nucleosome positioning and the chromatin condensation. So depending on how condensed is this, how nearby are each of these nucleosomes to each other, that will determine whether genes in that region are expressed or not. Right? and non-coding RNAs, right? So like messenger RNAs, silencing RNAs, all of this that we have in the genome. And yeah, so those are the four. And okay, so here is the nucleosome we're talking to you about. So the nucleosome is the basic unit of chromatin, right? Chromatin is what you see when you see these photos of the chromosomes, right? All of that DNA compacted there, that's chromatin, right? It's composed of an octamer of histones. So it has two copies of the histone H2A, which is the blue balls that you see there in the image in the top right. Then H2B, two copies of H2B, H3, and H4, right? Which are each of the proteins that you can see there. And around that, it's 147 base pairs of DNA, right? So they make like a loop around each of these proteins, and that is the basic unit of this chromatin. And so the compaction of these two between two of these units of the two nucleosomes, as I was saying before, it determines the gene expression. So depending on these epigenetic mechanisms that I mentioned before, these are responsible of how compact this chromatin is. And so you can have either heterochromatin, which is what you see in the bottom image there at the top, right? So it's a state that is really compacted and is not favorable to gene expression. So why is it not favorable to gene expression? Because then the proteins that are gonna come, like for example, DNA polymerase or RNA polymerase, when it wants to get there into a gene in order to synthesize RNA and then proteins, so that a gene can be expressed, it cannot enter that region because it's all compacted, right? So then those genes that are in that region that is compacted cannot be expressed and will not be expressed, right? But the other state is euchromatin, which is what you see in the bottom, right? It's a condensed state. So there you can see that there's some spacer. I don't know if this is troubling. There you can see that there are some regions in the DNA that where these polymerases can enter, and then they can read, and they can synthesize the protein, right? So then these genes will be expressed, right? There. Oh, sorry, I'm just gonna push this down. Yeah. And so, what I was mentioning about the post-translational modifications, you can have, for example, if I say that one of these histones that are, now you can see here better, like you have a histone here, for example, the H3, right? And they have little tails that, this is the histone tails, right? So, and it has we number each of the residues of, for example, lysine or arginine, for example, that are in this little tail, so that we can name them, and then each of these residues, they can have certain modifications. No, they can have methylations or acetylations. So, for example, if you have, let's say, the acetylation of lysine 14 of histone 3, of this histone, you would write it as H3, histone 3, K14, right? So lysine 14, and then AC, like acetylation, right? And this, for example, if you have this other modification, trimethylation of lysine 27 of histone 3, which is actually one of the most important modifications that can be related to cancer, the famous H3K27 trimethylation. You would write it in this way, right? So in the end, you will have Me as methylation, right? So that's our nomenclature there, so that you don't get lost in it. And now we go into which are the enzymes then, the proteins that are responsible of adding these little modifications to each of these histone tails. We have three types of epigenetic enzymes. So the first one are the writers. They're enzymes that are gonna go through each of the histone tails, and they're gonna stop at the residue, the amino acid residue that where it wants to put its modification, and then it's gonna put, for example, an acetyl group, right? So for example, that lysine residue is acetylated, right? Then there are others that remove modifications, which are called erasers. So these erasers do the opposite, right? They reach that residue and they say, no, I don't like it. I will remove that methylation from that histone, and then it's no longer acetylated, right? And there are also those that interpret and transmit these epigenetic signals that are called readers. So there are other enzymes that go and read which is the modification that you have there, and then that information then they can transmit back some other region to regulate then the gene expression of a certain region of interest, right? There's always a balance in here, like Thanos said, right? You have a lot of balance there with writers and erasers, or you should have this balance, but then in cancer, for example, as we will see afterwards, you have certain damages or modifications in these epigenetic enzymes, which may activate genes that you don't want, that may cause cancer, or it can don't allow some genes to express, for example, tumor suppressor genes, which also is related to cancer generation. So what is the importance of these mechanisms in cancer? As well, as I just to follow what I was saying. So you may have heard a lot about cancer. As I mentioned at the beginning, right? Everybody says, oh, you have these somatic mutations in the DNA that are causing cancer, right? But actually, epigenetic mutations, what we call epimutations, scientists have shown that they greatly exceed the number of genetic alterations that occur in cancer cells. So it may be even more important than the somatic mutations in the generation of cancer, because there are many more in cancer when you compare the same cancerous cell to a normal cell. Right? And these epigenetic alterations dysregulate certain processes related to cancer. For example, proliferation, cells proliferate more. Differentiation, cells stay into a stem cell state that allows them to reproduce a lot more. No? Tumor suppression, epigenetics cause our tumor suppression genes to not be activated. So you don't have anything to protect you from the development of cancer. And oncogenicity, for example, activating proto-oncogenes that are turned off normally in a normal cell, but now you remove that deactivation so that they can then cause the development of cancer, right? Oh, I have 10 minutes there. I have to go fast. So yeah, you can have then, as we were mentioning, some modifications in DNA repair genes, for example, which may also cause cancer to develop, or many others, right? For example, when you have methylation in the DNA, that typically occurs in the promoter regions, and so then that gene that should be expressed, for example, will not be expressed if you have hypermethylation of that region, right? And this will cause phenotypic changes, as you can see here below. Right? Cells will look way different when due to these epigenetic changes. Then, as I was mentioning before, scientists have discovered that that you can have mutations in these epigenetic proteins that I just mentioned before, and that those changes that may be, for example, a loss of function or a gain of function can also be related to cancer generation, right? So this is a way that we can have cancer. So what happens in what is the typical like what happens inside the cell nuclei when in cancer cells? You have a global hypomethylation, which means that in the repeated sequences in the DNA or the retrotransposons, which has their jumping genes, as we say, no, that can cause mutations and cause problems in the DNA, cause the instability in the genome, and it can cause the activation of oncogenes, which we may drive like excessive proliferation, for example, of cells. And you may, but in the other hand, you will have a hypermethylation precisely in the promoters of genes that you want to express in a normal cell, but in this case, due to their hypermethylation, they're not longer expressed. For example, in tumor suppressor genes, as I was mentioning before, so that you don't have any backup for tumorigenesis. And you can also have modifications in the histones that affect the compaction of the DNA. And so in order that the regions that have tumor suppressor genes are not expressed, and the regions that have oncogenes, they are expressed more than they should, right? Leading to cancer. And so the important thing and the thing that is promising for us is that these epigenetic modifications are reversible. Of course, as we will say, it's normal, right? You have these erasers that can remove these epigenetic marks, and they can be modified by specific chemotherapy treatments, right? Which is called epigenetic cancer therapy, which is this new advance that researchers are pushing for. And so what is the objective with this new therapy? It is to reprogram the phenotype of these cancer cells so that you induce their differentiation. So a cancer cell proliferates a lot. It resembles more like a stem cell, which is poorly differentiated. And because it's poorly differentiated, it's very difficult to attack with chemotherapy treatments. So what happens with epigenetic cancer therapy? You cause this cancer cell to become differentiated. And once it's differentiated, it's easier to kill, right? So then you pair epigenetic cancer therapy with normal chemotherapy, and you have a much more bigger success rate in your treatments. And this was very exciting for us in the lab last year when this was published, 2024. This was the first paper that finally proved that cancer can generate only with epigenetic changes, with epimutations, and without any need of genetic mutations, which was really exciting for us and something really good for our field. So in here, they mentioned in brief terms how a group of repressor proteins, epigenetic proteins, that's called the polycomb group proteins. In this case, it was the PRC1, polycomb repressor complex 1. When you ablate that, when something happens to that epigenetic protein, which would normally won't allow to oncogenes or proto-oncogenes to express themselves. So you could say that it's guarding you against cancer. When something happens to that protein, even if it's inactivated for a couple of days and then you remove that alteration, you have an irreversible activation of genes that are associated with cancer, which means that only this transient effect from the environment in your genome causes you cancer without the need of any other mutation in your DNA, which is incredible. This model was the fruit fly, Drosophila, but the polycomb group is very much conserved in humans. So this is very relevant to us. And so you see, for example, in here, when it's after this transient ablation of this protein, expression of JAK-STAT protein members that sustain hallmarks of cancer, right? Proliferation, cell growth, cell and metastasis, right? So this was huge for us. And so with this, then if you treat then with these epigenetic drugs that are classified into two classes, non-selective inhibitors and selective inhibitors. The difference between them is that selective inhibitors, you target the specific proteins or specific histone modifications in the genome to have an effect of interest, or you treat the certain proteins that you don't want in your to be expressed there in your related to epigenetic signaling. Then you can effectively or hopefully reverse cancer into a differentiated state, and then which will facilitate cancer in the future. So yeah, with this I finished. This is the recap of the session. So yes, so these epigenetic changes can be passed down. It depends. If you want, we can discuss that after I finish. These epigenetic changes can be heritable, and epigenetic changes can come from the environment, from pollution, your habits, everything, right? Then, as we said with that paper, like the modifications in the epigenome are sufficient and enough to induce tumor formation. And lastly, the epigenetic therapy can now is our new hope to revert cancer, and hopefully bring us with the missing weapon to get rid of this insidious disease that affects so many all over the world. So with this, I finish, and thank you for your attention. Any questions?
Thank you so much for sharing this. And it's really useful and valuable information. And thank you so much for also covering the basics in the beginning to make sure that people are, comfortable with going forward with it. So I do have actually a question. So for like the epigenetic therapies, are there some that are like available, and what would they choose to do? Say if they look at your cancer cells and they see that there's hypermethylation over here, then they would provide something that would help with demethylation. Is this like generally how it could work? Or...
Yes. So yeah, there are different drugs for that. Maybe I have some of them in my
Oh, and it's okay
slides.
if it takes time, then no worries.
Yes, in here you can see, for example, there are certain
Okay.
inhibitors, for example, here for DNA methylation. So yeah, the most widely used, for example, in here are the citabine, azacitidine, which target, for example, in here in the case of DNA, they target the here DNMT start means DNA methyltransferase. So it's the ones that enzymes that put this methyl group in the CPG islands in the DNA. So then, if you treat with these drugs, then you effectively inhibit their effect, and then you can, in this case, for example.
Hmm.
Awesome presentation, guys.
Thank you very much.
Yeah,
It's
great.
Nice to
Great
see
job.
you.
good to see you here.
So, like, I would just like to just relate to this as someone like I'm also working on epigenetic modifications and mutations. So, I would like to ask, like, is there any pre-screening test available to the patients, like, which we can see these mutations, or if the person is at a risk of developing cancer? Is there something going on around that?
Yes. Yeah, you can do some. I think I'm not. I don't recall exactly what's the name of or anything, but there's a test I think that you could look at your general, like for example, methylation levels. Yeah, at least in your DNA, I think you can do that. But yeah, like in terms of the price or what's the name, yeah, I will have to come back to you. But there
So,
is.
like, I'm
A
working on
way to do that.
epigenetic aging. So, like, for that, we have like some tests are available, but those are quite expensive, which can tell you about your epigenetic or biological aging. And from that, we can, estimate the risk of developing the cancers. but like you are working on specific cancers, and specific cancers may have like specific epigenetic mutations. So is there something like that, if we can, see if there's that particular mutation, epigenetic mutation or methylation over there, so that the person is at like a pre-cancerous stage, so we can identify it earlier and treat that, instead of just waiting for some time and see when it is developed completely and treating.
Yes. So yeah, there's well, in research at least, I don't know if that's approved for clinical use, but there's certain techniques. For example, that you have attack-seq. I'm gonna write it here in the so attack-seq, and you can also have chip-seq, which are techniques. For example, the chip-seq technique, which transfer chromatin immunoprecipitation paired with sequencing. You can look at the very specific histone tail modifications. So if I imagine if you take a biopsy from your patient or your animal of interest, and then you submit it to this analysis, then you will be able to see the presence of these specific modifications, where they are in your epigenome, like which genes is that modification affecting, right? And so yes, so you can do that. Or attack-seq is more general. I think it looks more into how the chromatin is compacted in the sample of interest. So you can have more idea of whether you have heterochromatin or euchromatin at certain regions in your genome of interest, right? So like if there's, for example, hypermethylation in an area that related to tumor suppression genes, that no, right? This person may be more prone to have cancer, for example, because of this specific epigenome state. So yes, you can do that using these techniques.
Thank you so much.
My pleasure.
So I wanted to thank you again. Also, Harsimran, it's a really beautiful question for screening. And I'm wondering, in some cases, would you necessarily need the screening? Because as you were mentioning, Federico, there's global hypomethylation as a pattern. So then if you were to give global, aid for methylation, then you might be able to reverse a lot of these conditions. Maybe, I don't know. But the thing is, there's also hypermethylation in certain spots, but it might be as a result to the hypomethylation. So perhaps if you were to simply generally activate methylation again of the DNA, so boost the enzymes that are doing it, don't just have a new enzyme that does it, but just boost the normal enzyme expression, uh, could it contribute to patterns of youthfulness? Maybe maybe it's not certain, or maybe it's too much of a simplistic approach.
Building on that,
But
I would, yes,
sorry,
sorry.
yeah, you go.
Yeah, so I was just saying, like, building on that, like, I'm working on aging. So we know these changes, these are not just because of, some disease or some like cancer. These are also because of your aging. Like with age, as we can see in the older age, there's global hypomethylation, which is similar pattern to the cancer one. So there's some new hypothesis that behind all this, the main reason or the culprit could be the aging. If we can stop the aging of that tissue, maybe we can, stop all the problems. Then
Well,
hypothesis, like, it could be interesting to see.
that is actually the hypothesis of one of our collaborators there in the lab. They attack cancer on the lens of aging, right? So they give anti-aging drugs that they call senolytics. So drugs that kill specifically cells entering senescence, because senescent cells, they create the senescence associated secretory phenotype, the SASP, which drives inflammation, drives all of these effects in the in the surrounding tissue where that senescent cell is, which is creates an environment prone for cancer, right? So, and cancer is a disease of aging, right? So most of the cancers like you can see like how the age progresses, and you have much more cancers in aging people than you have in the young people, right? So it's a disease technically accepted that related to aging. So yeah, like that's also an approach which if we can reverse aging, which is the approach, for example, of Brian Johnson that wants to live forever, right, with his diet and exercise interventions, whatever. Then you could basically, yeah, like two birds, one shot, right? So that's another important, approach.
Yeah.
Yeah.
Like now the people are saying like aging itself is a disease. So they're working through that perspective. So it's nice to see that.
Mm-hmm. Yeah, exactly. And yeah, Brian says we're gonna like effectively not put any more like death is not gonna be an endpoint, it's gonna be an option, right? So yeah, I don't know if they're gonna do it, but yeah, it's interesting at least what they say. And he does exactly, he does like methylation testing, I think, because methylation he relates like the scientists he works with also relate that to cancer, and so he sees that with interventions the methylation levels like go down, but I don't know like if it's I don't know how specific it is. I'm not sure. They just said that like my epigenetic age is lower because I have changes in this methylation, but like they don't say any specifics. So yeah.
There is a very famous researcher at Harvard, Dr. David Sinclair. So like he has worked a lot on aging and epigenetic clocks, and he has seen like with his precautions and the therapies. He says like his epigenetic age is like 20 years less than his chronological age. That's amazing, and you can see
Mm-hmm.
the influence of these things on your body itself. It's
Yeah,
great.
seriously. And thinking about it, like with methylation, I love the idea of methylation because it could get passed on from father and mother to child because it's strictly on your DNA. And I'm wondering, um, is this also potentially modifying our risk for cancer development in our lives? And are the things that we do in our life right now, like the stresses that we go through, times when we don't have proper sleep, is there like a predictable methylation change that happens and then it increases our risk? It's a hard question to answer. So like it's just something out of curiosity, and maybe it's just something to think about. But
Mm-hmm. Yes, so that's a very very important question, and yeah, my supervisor always told me like, for example, when you do a nuit blanche, like a all-nighter, right?
mhm.
Like the level of hypermethylation in your brain, it's incredible. Like it's night and day when you like
Really
after only one night of not sleeping, all of your brain is hypermethylated, right? So it's
interesting.
very changes are very fast, and that as you can see might be very dangerous because just transient change can give you cancer, right? So for example, yeah, I was looking at overworking and cancer, and so for example, other example is night shift workers have an altered DNA methylation, which is very similar to the level of methylation of early cancers, right? And also they have seen, for example, in these workers changes in so hypermethylation of genes like TP53, which
Oh, TP53.
produces the
Right, the tumor
P53
suppressor.
tumor
Exactly.
suppressor.
Yeah,
So
P53. Sorry. Yeah.
P53. And for example, there's mice in mice models where they put them not to sleep, then they have an increased tumor growth, and just because of that, right? And so what we can do in order to prevent cancer, like what measures we can take. Well, some are straightforward, like everybody knows it, but maybe not many do it. So healthy diet, right? No ultra processed foods, because ultra processed foods have, as we all have heard, right? These preservatives, colorants, emulsifiers, all of these substances that are poorly studied, and many of them have been linked with cancer. So we can take polyphenols present in fruits, in vegetables, for example, vitamin B in fish, for example, which have a great influence in some of these epigenetic enzymes. Well, I think there's a TET2, which is one epigenetic enzyme, very important, dependent of vitamin B. So your diet can help you prevent cancer. Obviously, no tobacco, as low alcohol as you can, because that causes epigenetic alterations in the tumor suppressor genes. Um, avoid exposure to pollutants. Filter your water, very important. We don't want any heavy metals in our water. Um, for example, don't consume a lot of tuna that has a lot of mercury in it and can also predispose you to cancer. Exercise, because why? Because exercise decreases inflammation and upregulates your immunity. If you have high levels of immunity, you can fight better cancer, right? Less chance of cancer. And avoid chronic stress. Like acute stress, well, we all have it. It's not, it depends on how you manage it. It can be even beneficial for you, but chronic stress is not good. It can also cause epigenetic changes that could lead to cancer. So yeah, this is my 2 cents on that. Yeah.
Absolutely. And the thing is, a lot of us, a lot of the students here, we basically do night shift work because like we're overworked on some phases. So then I didn't realize it, but I guess I have a lot of DNA methylation in my head. Um, yeah, like what do you think we should do about that? Like, maybe there should be like because I don't know if people are aware of it that like there's so much of a risk from over doing overnight work, and maybe people are going to be like. Maybe we should implement sleeping initiatives so that workers they have proper time to rest and recuperate. Okay, well, it's an open-ended question. It's there's many ways to respond to it, but yeah, let me know your thoughts on this, the DNA methylation with sleep and what we should do.
Yes, particularly in research is very hard, and even more so, I think in Canada, because I've seen colleagues working in, for example, United States, and like their, for example, the PhD program, I say that it's really treated like a job, so it's really 9 to 5, right? And that's the expectation. If you want to work more, like that's fine, but your supervisor, for example, cannot like force you, to like work overtime on staying, coming the weekends or whatever, which is, I think, like well, common practice here. Like it's like everybody does it, so like if you don't do it, you're doing something wrong, right? So which leads to overworking, stress, and potentially to cancer, right? So I think we could adopt certain things about the academic, like how academia is viewed in the United States. Like they have better wages for PhD, for example. Like if the supervisor tells you, oh, you have to work more, you can even go and there's a student association that can take those complaints, and then that can be passed down to the supervisor. You can have specific like established holiday breaks and sick days, like sick day leaves, which I found like fascinating. Like why don't we have that in Canada? Like it's a full-time job already, like even more than full-time job. So
Yeah.
why don't we have the benefits of a full-time job, right? So yeah, I think we could adopt certain aspects of that, but yeah, the household culture, I think is very ingrained even in the northern the North America mindset, which we should also find a balance, like not maybe not well, I don't know much about Europe, but I heard that, for example, some countries the life is much more relaxed, and the all of the like the work related issues, or if you want to go get something, like everything is very like chill, calm. That is also bad, because like if you want something urgent, you don't get it. Next month I will get it. So yeah, maybe adopt some things here and there, right? But yeah, we need to, yeah, have more discussions about it. Maybe send formal letters to deans of our faculties, and if we gather enough signatures, well, maybe we can start making a change, right?
Yeah, because it may not even be just the DNA methylation, or maybe there's modifications on the white blood cells as well, because white blood cells die sometimes when there's chronic cortisol. So maybe it's also linked with methylation of there should be an epigenetic change from cortisol that then makes your white blood cells die out and not work. So then you're not capable of getting rid of the cancer cells that you have because there's white blood cells that would normally take care of them, get rid of them. So then sleep for a number of reasons it would support us in this. I see there's another question. How
Uh, yes, go ahead, Kita.
Yes, hello. Thank you so much for this presentation. It was really interesting. So I don't have really a medical background, so my question will be more high level. It's more about a young population that tends to have, like, we have an increase of cancer amongst young people, so like lower than 50 years old.
yeah.
I think the median age is around 66, and now we see it more and more common amongst young people. So my question for you is like, what habits do you see might have, changed in our generation or younger generation that might cause this issue, and if ever we can prevent it on our own level?
I love it. That's an excellent question. And I also had the same question some months ago, and so I started reading a little bit more about it because yeah, as you mentioned, cancers are now coming down to younger and younger people. And for example, I was looking at the statistics, and there's a lot of a high increase. Mainly, well, of course, you're gonna have the prostate cancer in males. That's always be at the top, and breast cancer in women, right? Which is sex associated. But then the common denominator between the two is right beside that you have, for example, colorectal or like digestive tract related cancers. But and that people are having younger and younger, like that the amount of people that have that cancer, like in terms of age, you have a lot more in the young people. They say that is because of our habits in our diet. So it's mainly going back to the ultra processed foods and all of these pollutants on heavy metals in our food, in our water, mercury in our fish. So all of these, also well, the tobacco is related to lung cancer, right? But this, all of this contamination that is coming, that we eat, that we consume, is causing a really steep rise in digestive tract related cancers in young people. So what we can do is like try to go then with the or like I know it's more expensive, but it's worth it. Like it's your health, right? So try to eat as much organic food, vegetables and fruits as you can. That will be my suggestion. Use the Brita filters for your water, obviously, or some others that is it's not a paid promotion, but some others that remove all of these potential contaminants. And yeah, really watch what you eat. Don't eat too much ultra processed foods or something that has a really fluorescent colorants, because all of those are potentially carcinogenic, right? So yeah, it's just watch more what you eat, and given that there's also a really high peak in respiratory, like in lung cancer, is the top cause of cancer-related death in the world. So of course, don't smoke and don't be with other people that smoke, because secondhand smoke even cause there's a higher chance of having cancer from secondhand smoke than firsthand, right? So yeah, also watch with who you mingle with on a daily basis. That would be my suggestion.
Thank you so much.
My pleasure. Thank you for your question.
Thank you for sharing that. And if there are no more questions, perhaps this could be it. I'm really thrilled to see even after the Zoom cut off and everything, people were engaged to ask questions. Um, One last mini question: the colon cancer increase that you were talking about, do you think it's strongly associated with red meats or lipids from the red meats or things of this
Mm.
nature?
Oh, that's a tough one. Uh,
Maybe,
so red meats and fats, right? You mentioned.
yeah.
So mm,
Like if
I've
someone
seen.
has lean red meat, maybe it's not as bad. But
Uh,
I don't. Uh,
I mean, maybe because I think it depends on the origin. right? Because you know that you can have some like cows fed with a clenbuterol, I think. I don't know if that's what you say in English, or certain because then you feed, for example, these cows with a lot of hormones, like so that they grow a lot,
Right.
they have a lot of
Right.
fat, a lot of muscle, and it's more
Yeah.
delicious. But hormones, like they really mingle with our system, and as you can say, like everything that is external can have an impact in our genes, right? So there could be a possibility there, no, with that interacting of those artificial animal hormones with our genetic systems, but I'm not sure really. Like, I don't want to say something that I don't know. But there's also
Well,
other research that I have heard about the carnivore diet, right? That is like Andrew Huberman, neuroscientist at Stanford, Jordan Peterson, also in the United States. Is talking about like they after they adopted that, like they have seen such incredible changes in their positive changes in their body, right? They get sick less. They I don't remember if they have less risk of cancer, honestly, but like it could be well, maybe not beneficial, but better that because as we know, fats like eating fats and proteins is much better than eating a lot of carbohydrates, right? Because that makes you overweight a lot faster than those. And yeah, and then overweight relates to inflammation, to cancer, to and also glucose intolerance, insulin intolerance. So yeah, but I don't know. There's arguments on both sides, right? So yeah, I would have to look more into that. Yeah.
it's very interesting. And Harserman, you raised your hand.
Yeah,
Mhm.
on that point only. So like it's not like currently scientific like proven, like that's not for sure. But people say like eating carnivore diet or especially red meat, it leads to DNA methylation changes which are similar to those in aging. So people like,
Yeah.
but again, I'm saying like it's not proven yet. It's some basic studies, that's it. So, but they say there's some association with that.
Okay.
Mm.
Interesting. Thank you for sharing that. And yeah, it probably is the case. I don't think we have like evolved for too many years eating red meat as much as other forms of meat. So maybe it's just not as much tolerated. But yeah, another thing, mince meat, they add a chemical,
Mm.
an antioxidant to preserve the rich red color of the mince meat called propyl gallate. I had doubts that it was good for us, but I'm seeing quite a few research papers showing that this antioxidant that they add to the mince meat actually kills cancer cells, prevents cancer cell growth, but it's just the propyl gallate, not the mince meat. So they're adding
Mm.
things to the mince meat to preserve its color. It just happens to prevent cancer growth. So
Mm.
maybe there's high complexity of things. Sometimes you add a chemical, sometimes it seems to be fine. Sometimes you add a chemical and it's a problem. So it's about doing research to know the fundamentals of it. Does it actually help? Does it actually not? And a lot of times they're problems. But in this case, it was actually a beneficial one. But mincemeat itself may still be an issue.
Mm. That's interesting. Yeah. So yeah, they have arguments on both sides to process because mince meat, I think it counts as processed, right? So yeah,
Yeah,
like.
it's also very poor quality meat. Like you're getting a lot of lipid. So yeah, a lot of reasons to not eat it, but some chemicals they could potentially be of benefit. It's not even just one article. Like I'm seeing like quite a few. So yeah. Um, but thank you so much. Does anyone else have another question?
No, not for me.
Okay. Okay, so perhaps we could end it here. So once again, thank you so much, Federico. It was a real pleasure, and thank you so much for encouraging people to be more aware, and also, you basically united us to talk about cancer. And my apologies for the Zoom. I was going to give a reminder right at the end for them to come back, and then it cut off even earlier than what it said it was counting down to. So I didn't get to tell them to. Like it's not over. You could join the Zoom. So I apologize for that. But
No
thank you
worries.
so much for being here. It means so much to us. So thank you and have a wonderful rest of the day.
Thank you for your invitation, and thank you for everyone for coming. That means a lot.
Thank you so much.